Good morning, class. We are going to talk about Chapter 11, Physical and Chemical Control of Microbes. What is contaminating? It is defined as any unwanted microbe. When you do your experiments in lab, and especially when we get to pure culture, and you realize that you have more than one microbe in here. One might be yellow and the other might be green. Well, you have at least two different microbes in there, colonies for it. So if you were trying to do a pure culture, just one, it would not be wanted. It's an unwanted microbe. I don't think this video plays. Why do we need to control microbial growth? To prevent spoilage of food and disease, we need to use the aseptic technique, which is free of all contamination. You've heard the term aseptic technique. You've done an example, a lab online. We're going to do another one today, and we're going to be using the aseptic technique the rest of the semester to do whatever we're going to do when we culture bacteria. Okay? There are two types of control methods, both physical and chemical. Physical and chemical controls are used to kill or inhibit. That says inhabit, but it should be inhibit the growth of microbes. Primary targets of microbial control: microorganisms that can cause infection or spoilage that are constantly present in the external environment. Contaminants that need to be controlled: bacterial vegetative cells and endospores. Endospores are so resistant, the goal is sterilization. Fungal hyphae and spores, yeast, protozoan trophozoites and cysts, worms, viruses, and prions. What is a prion? Come on, say the whole thing. Yes, okay. Anybody ever had a yeast infection? Don't know what that is. Okay. Now, though these may be some videos that you can look at on your phone or on your device, but I can't play them here. Why do we need to control microbial growth? To prevent spoilage of food and disease. Same thing we just did. Two types of control methods. What are the two types of control methods? Physical and chemical. The suffix "side," when you see C-I-D-E at the end of a word, it means to kill. Like a bactericide is a chemical that destroys bacteria, but not endospores. A fungicide is a chemical that can kill fungal spores, hyphae, and yeast. A virucide, a chemical that inactivates viruses. It didn't say kill viruses because our virus is alive without the host. No, but it inactivates them. Sporicide can destroy bacterial endospores. The most resistant form of a bacteria is its endospore. Endospores are much harder to kill than bacteria themselves. A So the only thing that can kill bacterial endospores is a what? Sporicide. A germicide and a microbicide are chemical agents that kill microorganisms. So you need to know the difference in side and stasis. Stasis and static, same thing. It means to stand still or inhibit. So a bacteriostatic prevents the growth of bacteria. A fungostatic inhibits fungal growth. A microbistatic, materials used to control microorganisms in the body, for example. So if I have a bactericide or any agent that is used on viruses, spores, whatever, it ends in what? Side. You may see a sidal form of it, but C-I-D-E and C-I-D-A-L, same thing. And it means to kill. But the difference in it, this is on the quiz on 1112, static or stasis only means to inhibit the growth of it. It won't kill it, but it won't keep growing. Does the application require sterilization or is disinfection adequate? Name some things that, let's just say, physical control, that we definitely want to be sterilized. When I go to have a surgery, like when I had my foot surgery, do I want that surgeon's instruments to be sterilized? Yes. Can you think of something else? When processed food, the environment in which it is done should be sterilized. Is the item to be reused or permanently discarded? When we use our tubes over in lab today, they are to be reused. When we get through with them, the lab coordinator will wash them out, then put them in the autoclave machine. That dry heat will cause them to be sterilized. If it will be reused, can it withstand heat, pressure, radiation, or chemicals? Is the control method suitable for a given application? Will the agent penetrate to the necessary extent? Is the method cost and labor efficient, and is it safe? It may be cost and labor efficient, but is it safe for humans? What is microbial death? When various cell structures become dysfunctional and the entire cell sustains irreversible damage. If a cell can no longer reproduce under ideal environmental conditions, death begins when a certain threshold of microbicidal agent is met and continues in a logarithmic manner. Give me an example of a logarithmic manner. 2 to the 10th power, 2 to the 5th, logarithmic manner. That's what it's talking about. Exponents. Factors that influence the action of antimicrobial agents. The number of microorganisms, just simply how many are there. And everything that's up here is also where? On Blackboard, under what? Course documents, chapter 11, PowerPoint. Okay? The nature of the microorganisms in the population, the temperature and pH of the environment, the concentration of the agent, the mode of action of the agent, and the presence of solvents interfering organic matter and inhibitors. How do these antimicrobial agents work? The cell wall, first thing it does, it tries to break down the surface of the cell wall. It blocks its synthesis. In other words, synthesis means to make more, synthesize more cell wall. It tries to digest the cell wall, break down its surface, and then after it does all that, the cell becomes fragile and is lysed. What does lysed mean? Break, okay? Then it takes action on the cell membrane, protein and nucleic acid synthesis, So if you can't synthesize proteins, if you're a human, you do what? What happens when you can't synthesize proteins, when you don't have ribosomes that... And you can't make protein, like the case of a gentleman who used some kind of an umbrella to shoot somebody with some ricin. I'm just passing by you now, and I hold my umbrella up and shoot some ricin, R-I-C-I-N. Ricin inhibits, it stops protein synthesis, and you die in 3 days. Okay? Protein synthesis, when you can't synthesize proteins. And likewise, nucleic acids, which is your DNA, RNA. But in bacteria, it has a very similar effect. Protein synthesis, it stops it. It binds to ribosomes to stop translation. It binds irreversibly to DNA, preventing transcription and translations, and it has mutagenic agents. Controlling microorganisms. General considerations. These words are most important. Most students understand it when we go through it, but then on the exam, I get a different idea about, did you really understand that sterilization is the destruction of all microbial life? Your surgeon's instruments that he uses on you in surgery, there should be no microbes on them because they've been sterilized. Can I sterilize my skin? No. Sterilization only takes place on what kind of objects? Animate or inanimate? Inanimate. So can I sterilize this desk? It's an inanimate object. But I am an animal. I am an animate object. And I cannot sterilize my skin. Disinfection destroys most. So look right there at that difference between sterilization and disinfection. Sterilization destroys all microbial life. Disinfection destroys most. It's a difference in all and most. Reducing contamination on what kind of surfaces? Inanimate surfaces. So can I disinfect my hand? No. Antisepsis destroys most microbial life, and it reduces contamination on what kind of a surface? Living. So can I apply an antiseptic to my skin and destroy most microbial life? Yes. Name some antiseptics. Say it loud. Some kind of powder, what'd you say? Yeah, okay. Hand sanitizer could be an antiseptic because it's got what in it? Alcohol. Name some more antiseptics. Huh? Hydrogen peroxide is an antiseptic. Okay? Decontamination is the mechanical removal of most, again, most, microbes from an animate or inanimate surface. And it's also called sanitization. You see it written there as sanitization, and McGraw-Hill has it written both ways as sanitization on some slides, and then it turns around and says sanitation. They mean the same thing. And degermation means the same thing. So of those different methods of microbial control, which one can be used on an animate or inanimate surface? Decontamination. Facts about physical control. Heat, sterilization as an agent of microbial control. Heat is used. Temperature and length of exposure must be considered. Higher temperatures generally allow shorter exposure times. Lower temperatures generally require longer exposure times. Two more key points. It's on the quiz and it's on the exam. thermal death time and thermal death point. So what is the difference? Thermal death time is the shortest length of time required to kill test microbes at a specified temperature. We're talking about heat here. But the thermal death time is the shortest length of time required to kill all test microbes at a specified temperature. Thermal death point is the lowest temperature required to kill all microbes in a sample in 10 minutes. Kind of get the difference in those? Thermal death point. What temperature is required to kill all these microbes in 10 minutes? Oh, I'm sorry, excuse me. Let me know when you have it. All right. So for these methods of control, we have the definition, the type of control. Everything we're talking about right now is physical. We haven't talked about chemical controls yet. So a method of control is sterilization. It's used on inanimate objects. It removes all microbial life. If it's sterilized, there is not a microbe left on it. Filtration, which removes or traps microbes, and it's not a sterilization method used on inanimate objects. Pasteurization kills potential infectious agents, spoilage, and it gives sterilization. And pasteurization is used in beverages. Name a beverage that is pasteurized. Milk. Who discovered or came up with the process of pasteurization? Pasteur. Autoclaving is used to sterilize its heat under pressure. Inanimate objects. Radiation. We have different types of radiation. We have ionizing radiation and non-ionizing radiation. Non-ionizing radiation is radiation like ultraviolet rays. Ultraviolet. Where do we get ultraviolet rays from? The sun. Can I get as much ultraviolet non-ionized rays if I am on my back porch as I can on the beach? You don't think so? My back porch doesn't have any trees inhibiting the sun. So when I lay out there on the deck, it's just like I was on the beach. Put it in your mind like that, unless you like the beach. And then you'd rather be on the beach. All right, drying and ionizing would be like X-rays, gamma rays, cathode. What is the name of the female who died as a result of too much radiation? She discovered the structure of DNA. Say it. Who? I can't hear the time you said Vivian. No. They took a what? Oh, you're thinking about, yes, that's who I'm talking about. Her name was what? Rosalind Franklin. Rosalind Franklin. Why did she die? Okay, can you have an X-ray every day? No. What happened to you? All right. Okay. Henrietta Lacks. That's who you talking about. It just came to me. But her family finally did get some money for it. Finally, after they've been selling her cancer cells for decades, and her family got nothing, she died, but finally, she got some justice. There is justice. It just might look like we might not ever see it again, but in this country. Okay, keep going, Janice. Drying or desiccation is another method of control. It's a process which makes things extremely dry by removing the water. What are some desiccated foods that you like? It's not desiccated, it's not dried. I like dried bananas, dried pineapples, cranberries, okay? Drying or beef jerky. I don't like beef jerky, but some people do. That would be okay. Facts about chemical controls used to interrupt the spread of microbes. No single chemical agent is ideal for controlling all microbes under all conditions. Chemical controls are not effective on bacterial endospores, spores, or cysts. So here are some chemical controls. The method, disinfection, reduces contaminants. It inhibits or destroys them. It's used on inanimate objects. So those physical controls, what were they? Heat. And in talking about heat, I want to just go back for a minute. There are two kinds of heat. Dry heat, moist heat. When I boil water, if I had a grandbaby, I would boil the bottles. Would that sterilize my bottles? It would not. If I would put them in a pressure cooker, heat under pressure sterilizes. Just boiling the baby's bottle will do what to it? It won't sterilize, but it'll do what? Disinfect it. Okay? All right, some chemical controls. Back to this. Disinfection, we know. Antisepsis removes microbial life on living tissue. That's the big difference. You see, it's in red. I didn't do this PowerPoint, but the person who did wanted to really drive home that idea that antisepsis is on living tissue. Decontamination removes most microbes to safe levels. It is considered a chemical and physical control, and it's done on inanimate objects. Degermation reduces the number of microbes on the human skin through scrubbing. It's chemical, and it's used on living tissue. Sanitization, cleansing technique that reduces or removes microbes. It's used on inanimate objects. So a chemical that's used to treat a disease is called a chemotherapeutic agent. Chemicals that kill microorganisms are called germicides. Chemicals that inhibit microbial growth are called germistats. Germicides used on inanimate objects are called disinfectants. When germicides are applied to living tissue, they are called antiseptics. We got that now, don't we? You can take that quiz right now, couldn't you? But you got that point. Will you miss what is sterilization, disinfection, antisepsis? You got that? Factors that affect the germicidal activity are chemicals. The nature of the microorganisms being treated, the nature of the material being treated, the degree of contamination, the time, how long, time of exposure, and the strength and chemical action of the germicide. Some germicides are phenols, alcohol. What's an example of a phenol that denatures the protein? Lysol. Alcohols disrupt lipids and denatured proteins. An example is ethanol or isopropanol alcohol. Halogens inactivate proteins. What are some halogens? Iodine, chlorine, hydrogen peroxide. Surfactants damages cell membrane and activates proteins. What's an example of a surfactant? Ammonia, detergents, soaps. A little earlier, someone thought that soap was an antiseptic, but actually it's a surfactant. It inactivates proteins and damages the cell membrane. And alkylating agents like alkyl groups, and an example is ethylene oxide. Preserving foods. Food spoils only when microorganisms grow on them. Preserving food is largely a matter of eliminating microorganisms or slowing their growth. In food preservation, we have to consider the temperature. Now, on one of the quizzes, I had a question on there about, they had a class picnic. LaKindness brought some pasta salad to the picnic. While they were out doing something else, the pasta salad sat on the table for 3 hours. What are some things that, and she started out introducing 20, because this might be on the final. She introduced 20 microbes into this pasta salad. And if it sat in the doubling time, how long does it take another generation of microbes to make? Does that make sense? So we call it doubling time. And if the doubling time is 20 minutes. So if 20 minutes from now, if you started out with 20 microbes, 20 minutes from now you got 40. Okay. So if it stayed there 3 hours, how many 20-minute periods are there in 3 hours? 9, because every hour got 3 times 20 minutes is 60, right? So, I gotta find something that rhymes. You got an expo? Okay, good. Thank you. I appreciate you. You ready? Very prepared.
I got a board in my house.
Yeah. Oh, you got a board in your house, so you have to have these. Okay. All right. So that pasta salad had 20 microbes to start with. And then the doubling time. Doubling is how many? 2 to the what power? 9th power, because we said that there were 9 20-minute periods in 3 hours. Right? So, how many microbes exist in that pasta salad at the end of 3 hours? Anybody got a calculator care to do the calculation? For a bonus question that might be on the file? It was on the other student quiz. 2 to the 9th. You have to have a calculator with the exponent function and do it, Miss Ma'am. I'm going to save you a little whoa. Somebody tell me. Yeah, 10,000 what? But you said 10,000. Yes. 10240. So you see how microbes can get to be so many? So what were some things that could be done to prevent it from getting all those microbes on it? First thing, she set it on the table. So what should have been done with it? Yeah, refrigerator. If you had a refrigerator, that would be the ideal thing, put it in the refrigerator. I don't know what kind. Thank you. I don't know what kind of utensils she used, but the utensils should have been cleaned. She should have washed her hands. Maybe she did. I don't know. I just know that at the end of 3 hours, letting it sit out. That's what happened. pH involves, okay, so back to temperature. Refrigeration was the first thing, then canning, pasteurization, 63 degrees Celsius for 30 minutes or 72 degrees Celsius for 15 seconds. See how the higher the temperature, the shorter the time it takes to pasteurize. You see that? 72 degrees Celsius is higher than 63, and the difference is half. No, less than half. 15 seconds is what? 1/4 of 1 minute? But it takes 30 minutes at 63 degrees Celsius. pH, they use vinegar, lactic acid fermentation. Then water, drying, evaporation, and freeze drying, and salting has a high osmotic strength. Chemicals that are used as preservatives. So what are contaminants? Any unwanted microbe. Why do we need to control microbial growth? Yeah, we want to keep it from spoiling food. And definitely we don't want any microbes growing on something that should be sterilized. What is wrong with this statement? Prior to vaccination, the patient's skin was sterilized with alcohol. Right, you can't sterilize skin. Alcohol as an antiseptic may have been applied, and that would reduce the number of microbes, right? What would be used to correct the wording? Skin was what? Can I disinfect my skin? I could decontaminate it, or I could say that, change it around somehow to use the word antisepsis or antiseptic. See there? We finished chapter 11. Does anybody need a 5-minute break, or can I do 12 if I promise? Hold on a second. Whatever. Oh, right.